So yeah, over the summer, we explored how plant defenses downstream of mediator unit 16 regulates plant resistance to turnip mosaic virus and its aphid vector Myzus persicae. So that is also a lot to unpack. I will jump right into the background. So turnip mosaic virus is a protevirus, which are positive strand RNA viruses that currently account for about 30% of all plant viruses we see today. And so these viruses are transmitted by aphid vectors. And so being able to understand how these plant aphid interactions work would be super helpful in developing pest management techniques, especially in an agricultural setting. So the vector virus interaction that I'm going to be talking about for the rest of this talk is between Myzus persicae, also known as the green peach aphid, as shown on the bottom left picture, and Turnip Mosaic Virus, or TUMV, as shown right here. And so currently TUMV is one of the most destructive viruses to vegetable crops worldwide. And so being able to understand these plant aphid interactions, we could use it to our advantage, in the future to perhaps even increasing vegetable crop output. And so the picture on the bottom right-hand side shows a TUMV GFP tag infected Arabidopsis thaliana, which is the model organism that we used over this summer. And so this plant is under UV light, as you can see from the purple lighting, and the green light that you see being fluoresced in the middle is where the TUMV is being expressed in the plant. So a little bit more about TUMV. We know that it is a protevirus, but it also encodes a polyprotein, which is cleaved by 3 of the proteases it produces, P1, HC-Pro, and MIA-Pro. And down here is an image of the genome, and the 3 proteases are highlighted in red. And so these proteases cleave the polyprotein to create 11 separate proteins. So in order to figure out what the significance is of every one of these proteins in TUMV to aphid fecundity, or the aphid's ability to create offspring, previous researchers in Dr. Castillo's lab cloned each one of these major proteins and transiently expressed them in host plants, in this case in enzyme. And they found that only the presence of one of these proteins significantly increased aphid fecundity when it's compared to the empty vector. And that protein here is under the blue bars, MIA-Pro. And so the image on the left is a picture of the bioassays being done on NBEN. And here, one aphid is trapped underneath these cages. And then after 12 days, they were counted in order to determine the change in fecundity. So knowing now that MIA-Pro is significant in somehow increasing aphid fecundity, the virus vector host interaction is becoming a little bit more clear. Now we know that TUMV is coding for this specific protease named MIA-Pro that's somehow altering plant chemistry, and that is somehow increasing aphid fecundity. What we focused on this summer is to figure out how MIA-Pro itself is altering this plant chemistry. So previous researchers, Manisha and Aureli, wanted to verify that this increase in aphid fecundity wasn't just plant specific. So they repeated the bioassays in Arabidopsis thaliana. And what they found was that in the presence of MIA-Pro, aphid fecundity did increase. So to further investigate the proteolytic activity of MIA-Pro, they also created MIA-Pro protease mutants. And what they found was that aphid fecundity was significantly reduced. And this is also another picture of the aphid bioassays being done, but this time on Arabidopsis. And so since we know, if you guys remember from the TUMV genome at the beginning of the presentation, MIA-Pro is a protease. So that got us wondering whether maybe MIA-Pro is cleaving some kind of plant protein that causes this downstream effect of aphid fecundity increasing. So in order to investigate this further, a researcher from Dr. Castillo's lab has done a bioinformatics screening of the Arabidopsis thaliana and Nicotiana pentamiana proteome for MIA-Pro cleavage sites. And he searched for one of the most predominant cleavage sites, which is the VXHQ site, and he found hundreds of plant proteins that contain it. In Arabidopsis, as you can see here, there's 625 genes, and in Nicotiana pentamiana, he found 1,293 genes. And so one of the proteins that we found from this screening is called mediator subunit16 or MED16 protein. And we thought that this was specifically important because it, or interesting, is because it mediates the crosstalk between the jesmonic acid and ethylene plant defense pathways. And as you can see here, MED16 does play a significant or a key role in inducing those downstream pathogen defense responses. And so with all this background information, I bring you to our hypothesis that we explored this summer, and that's that TUMV infection increases aphid fecundity by somehow altering the MED16 protein. So in order to do this, we had 4 different groups of Arabidopsis plants. One is the mock plant. The second one was the mock plus aphid, and then a TUMV infected plant, and then TUMV plus aphid infected plants. So what we did with the healthy plants, or the mock ones, is that we rub-inoculated them using tissue from healthy Myzus and Samiana, which is our source plant. And then for the TUMV plants, we rub-inoculated them with tissue from the TUMV GFP infected Myzus and Samiana. And so after about 9 days post-inoculation, which is when the TUMV starts to show symptoms of infection, we caged on about 20 aphids onto these leaves, and we let them feed for about 48 hours. And so on the picture, on the right-hand side, you can see on the top is TUMV infected envens, and on the bottom is TUMV infected Arabidopsis under UV light post-rub-inoculation. So after 48 hours of the aphid feeding, we removed the aphids and collected tissue. And from that tissue, we did some RNA extractions, and then from there, we synthesized cDNA in order to measure MED16 transcript levels using a quantitative real-time PCR. We also did protein extractions, which we ran through polyacrylamide gels, and then did Western blots with them. And so on the left-hand side, you can see an image of the real-time system machine that we used to run the QRT PCRs. And on the right-hand side, you can see the polyacrylamide gel chambers that we use to run the gels and then do Western blots with. So from this methodology, you can kind of see how we're seeing or exploring how TUMV infection affects MED16 both on an mRNA level and also on a protein level. So from the quantitative real-time PCR, we found that with TUMV infection, there was an increase in MED16 transcript abundance, which you can see from the right two bars. Yeah, right two bars as compared to the mock and the mock plus aphid. And so from the Western blot itself, we also do see an increase in MED16 protein and also a cleaved MED16 product in the TUMV and the TUMV plus aphid plants. So to speak a little bit more about this MED16 product right here, we look closely at the NeoPro cleavage sites in MED16 in Arabidopsis and Myzus and Samiana. And this top chart right here was done by my amazing mentor, Dr. Swante. And so one of the cleavage sites that we found interesting was the VLPEV site right here that is bolded. And if you look at the cleaved protein sizes, we have 111.7 kilodaltons and 29.3 kilodaltons. And this is interesting because the sizes correlate to the size of the cleaved MED16 product that we saw in our Western blots. So this may suggest that, oh, well, maybe it might be NeoPro from TUMV that's cleaving MED16. And so we decided to also focus more on NeoPro and not on the other two proteases that were encoded for in TUMV, because if you look at the cleavage or known cleavage sequences for HCPro and P1, they weren't really found in Arabidopsis or Myzus and MED16. We did find slightly close sequences to the known sequence, but having those amino acid substitutions are very rare, or the probability is very low. So to bring everything back to a full circle, my mentor also got this data by doing further bioassays in which he did, investigated how aphid fecundity and bioperformance changes in MED16 mutant plants. And what we found was that Aphid fecundity was actually higher in the MED16 mutant plants, as well as the percent of infected plants were higher. So this goes to further suggest that, well, MED16 might be a key player in plant resistance to UMV. So to conclude, or as a summary to all the results that we got from the summer, UMV infection does induce MED16 transcripts in Arabidopsis. And this makes sense because if MED16 is perhaps being cleaved by something in UMV, then the downstream response of pathogen defense would not be triggered, and this would cause the pathway to try to create more MED16 transcripts in order to get more MED16 protein. And so we also found that UMV infection increases MED16 protein and MED16 products, or the cleaved MED16 products. And from the bioassays, we also see that MED16 is required for resistance to aphids and to UMV. And so bringing back the viral vector host trifecta from the beginning of the presentation, the sort of trifecta interaction is becoming a bit more clear now. Now we know that UMV is encoding for a protease called MEDIATOR that probably is maybe altering plant chemistry by cleaving this protein called MEDIATOR SUBUNIT16, which is a key player in plant defense systems. And this is somehow increasing viral performance in plants, which also increases aphid fecundity. However, a lot of more experimentation needs to be done to confirm that it is just MEDIATOR cleaving MED16 and causing all of these downstream effects. And so this brings me to the future directions portion of this talk. And that so in order to confirm this cleavage, we can cross MEDIATOR with MED16 fly in Arabidopsis. And we could also confirm this cleavage in Nicotiana benthamiana by transiently expressing MED16 with MEDIATOR using co-infiltrations. And so that brings me to the end of my talk today. I just want to take a moment to thank my PI, Dr. Castillo, for allowing me to do research in her lab this summer, being such a great PI. Dr. Ray for being an amazing mentor and answering many questions. Manisha, Aureli, and Steve for without their preliminary data and research, I would not have a background to share with you guys. And also PTI and Delaney, Olivia, Adrian, Susie, and the rest of the interns for the great support and the great times that I've had this summer.